I would love to introduce you. So next, we are going to hear from Dr. Immanuel Lerner, the CEO and co-founder of Pepticom. Dr. Lerner is a scientist and biotech entrepreneur with a proven track record in envisioning and establishing AI-biotech startups and projects. He co-invented, founded, and is leading Pepticom, a disruptive company in the field of AI-based peptide drug discovery and design. Dr. Lerner led Pepticom from concept to revenue generation via collaboration with large multinational companies, negotiated and executed discovery deals, and also seed and round A funding for finance. Dr. Lerner is also a biochemist with 15-year academic prior to the experience academic prior to the establishment of Pepticom, and includes a PhD in biochemistry, followed by postdoctoral research at the Hebrew University of Jerusalem. During his studies, he published research in leading journals such as Blood, Nature and Communications, the Journal of Clinical Investigation, Cancer Research, and others as well, and had the honor of publishing his postdoc research with the 2017 Nobel laureate in Physiology Medicine with Professor Michael Rosbash. That is quite impressive. Thank you so much, Dr. Immanuel, and over to you. Looking forward to hearing what you've got to say.
Thank you, Roxy, for the introduction and the ability to speak in this very interesting event. And the rest of the former speakers were really excellent and a very good introduction to right into what I'm going to say. So Pepticom is into really the discovery and optimization of peptide-based drugs. And I will talk about the need for incorporation of AI into the drug discovery and development in peptides specifically. So you may know that we are celebrating the 100th year of insulin, which saved millions of lives. And it's basically a large peptide. The peptide is basically a short protein built from a string of amino acids. And as you may know, most of the peptide drugs on the market are either hormones or derivative of hormones. But in the last 10 years or so, we are starting to see completely synthetic peptides getting into the market as potential drugs. And this is changing the peptide market dramatically. It is very challenging to bring this completely novel peptide because you have to have a way of discovering these peptides. In a very short, I would say that the peptide drug market is projected to double in the next 5 years to something around $51 billion due to these new medicines. So thinking about discovering a new drug and you don't have a hormone to start with or a native peptide to start with, you need a screening method or a discovery method. So a breakthrough was done and the Nobel Prize was given to phage display, which is basically building a huge library, physical library of 10 to the power of 9 peptides. I won't get into too much details, and basically throwing them on target and getting an active peptide, and then with very hard work, with a lot of time and effort and cost, optimizing it to a drug. And after this breakthrough came another breakthrough, which was the invention of basically what is called mRNA display technologies, which is an elaborate way of doing something like a phage display with 10 to the power 30 molecules without a phage, which is a very impressive technology. And this also enables the incorporation of non-natural amino acids, which is very crucial in many cases in peptide drug discovery to make the peptides more drug-like. So this brings us to the current stage of where biotech and pharmaceuticals are as far as discovery. And what you can see here on the screen is a successful discovery project. And as you heard before, discovery and development of drugs in general and peptides is still a very inefficient process. 85 cents lost to each dollar put into R&D, and around $2 billion of drug development and discovery if you take into account all the failures. So this is a very good story. This is a peptide that is in phase 2 from Merck MSD, which is an orally available, fully synthetic peptide. So this is not a derivative of any known peptide in our body that was discovered using a display technology. In this case, something that came out of Pharmaceutical, collaborating with Merck MSD. And what you can see here, and that the reason I'm putting this, first of all, this is going to really drive the peptide market, but the time that it took to discover, from discovery to an early hit, was 3 years and $4.5 million, and then another 5 years and $12 million, just for the chemical optimization and discovery of the molecule. This doesn't take into account of all the other costs around. And when you think about that, that's really a very, very long time. And there really is a need to try to make this process more efficient. So why is it so difficult to discover peptides using these methods, which are really amazing methods as far as the biochemistry? So there are a few very basic things. First of all, you are talking about a chemical space which is around 10 to the power of 30 different solutions. And these techniques are screening only 10 to the power of 13 solutions at best. So they're not screening not even a millions or billions of the real problem. And the second thing is, once you have an active peptide, because this is a chemical library, you have no idea where this thing is actually binding. So it's very difficult to optimize these molecules to drug candidates. So you spend years in trying to understand how the structure activity relationship basically in these cases. And this is really time consuming and very, very cost ineffective. And the third thing is the fact that when you're using a library, you don't really control almost anything. I mean, you put the natural building blocks of peptides, you may incorporate a few non-natural amino acids, and you know what the size of the peptide is, but you don't really control any other aspect. So you can have a very good binder which has a horrible solubility. This was briefly discussed by one of the speakers. And all of the above are things that, at least in concept, can be addressed by artificial intelligence. So what we want to do, and what the direction the world is heading to, is basically moving from hormones to what we have today, which is library-based new drugs on the market, to drugs that are basically designed via artificial intelligence. This would enable, as we heard, and more effective time management, reduction in cost and risk, basically. So the way we believe in Pepticom and the way forward in our case of new peptide drugs is using reinforcement learning. And we are doing this because of many different reasons. One of them is the kind of data that is available today. Taking into account that data on cyclic peptides, and we are talking about small cyclic peptides, you can't really build on big data here, because there isn't a lot of information or data around these kinds of molecules. So we choose reinforcement learning. Basically what we do is we take a structure. It can be a crystal structure, it can be, if there is a good prediction from AlphaFold, it can be a prediction. We use our algorithms that were developed in Pepticom, and then we basically do an in silico discovery process where we have different strategies trying to find the optimized molecule. And once the run is over, we synthesize the peptides and we actually test them in-house and validate them and then optimize them. When we started a few years back, our first publication, that was an academic collaboration with Oxford University and Hadassah Hospital in Israel. We had a very primitive version of technology. We had only natural amino acids and linear peptides. We got very nice results. I won't get into the paper, but we found a very interesting peptide that could tackle an ultra-orphan disease. But we understood very rapidly that this is not good enough in order to compete in the peptide drug field, where you need really superior attributes for your molecule. So we started working on cyclic peptides and heavily non-natural modifications of these peptides in order to give them the right qualities they need. And we got to a point today where we have a very, very diverse chemistry, when we can control basically a lot of aspects of the physiochemical aspects of the peptides that we discovered in our AI software, basically, which is a very good situation to be in. And just as an example, you can see here in red the 20 natural amino acids, natural building blocks, but there are hundreds of other building blocks that most of them can be used in drugs that are, in most cases, very hard to incorporate into screening methodologies. And we have basically programmed 300 different building blocks. So we have a much more diverse chemistry basically to play with when we try to discover these new potential molecules. Another aspect, obviously very important, is the ability to predict the outcome. And we are working on that as well, but I won't get into too much details here. So as I showed you before, we are working with chemistries of cyclic peptides, non-naturally modified. This enables very active peptides, and I will show a few examples in a minute. One of the outcomes of these chemistries is very stable peptides. You can see this bar graph where our peptides are extremely stable in human serum. One of the known problems of peptides is their poor stability, which is a very bad starting point as far as a drug. So this is just an example. We have many other examples. We run our software on AWS. It's basically an ephemeral supercluster that can run parallel projects in the same time. And here is an example of a project. We focus most of our activity on in-house projects, but we have also collaboration with large companies. So this is a project where we started a very, very challenging protein-protein interaction inhibition on an autoimmune disease where there's a real need for new therapies. And we started in the left. This is an inhibition test with a hit molecule. And you can see that as we go to the right side, the potency of the molecule increases. So these are iterations on the molecule. And then we were able to reach a lead potencies as far as the biochemistry. And this is a very good indication to the fact that our model is correct, because if you have a wrong model, you'll never be able to optimize a molecule unless you synthesize thousands of molecules. And we did, you can see here, some other tests, and this is now tested in mice in this auto-inflammatory model, and we got very, very nice results that basically are in par with standard of care. And we have other interesting aspects of this molecule, but I won't get into more details here. This is another example where we took us 3 months to discover a peptide, which is basically an inhibitor of the SARS-CoV-2 interaction with the human receptor and basically cell entrance. Bear in mind, these molecules are completely synthetic, so they don't look anything like a short version of one of the proteins involved. These are completely synthetic, novel peptides. Our aim is basically to improve the technology and bring it to a stage where we need to screen less than 100 peptides in order to start from scratch, from an idea to a very highly active molecule. Obviously, there'll probably be a need to play a bit more with the chemistry, but really bring a valuable molecule that can be used in the pharmaceutical or in the biotech industry. as a very promising starting point, because it's very difficult to bring like a hit which is not extremely potent and you don't know how it works. So you need this validation, and this is exactly why we do a lot of work in-house. And basically the company itself is a mixture of computational chemists, software engineers, and biochemists trying to work together and look at the project as a whole from a holistic point of view, because we believe that there is really the need to connect the dots and bring something, in order to bring something that has value in the industry. So this is why we work in such a way. It's very challenging, obviously, to have such a multidisciplinary approach. And we are very fortunate to have two really key persons, key opinion leaders in the industry, Dr. Walid Danho and Dr. Tommy Sawyer. Both of them actually discovered and developed drugs that are in the market today. So they give us a lot of insight about how to do this in a correct way. And that's it. Thank you very much for your time.
Thank you very much, Dr. Lerner. That was really interesting. And I think there are a few points that I want to touch upon, but we'll do that during the panel session, if that's okay. And again, please do go onto the chat and have a look for Dr. Lerner's contact information, his LinkedIn, and also details for Pepticom as well. It would be great for you to engage directly with his posts, and I can see that you are actively promoting this conference as well. So thank you very much for that as well.